As the voice just said, this meeting is being recorded. Our guest today is Dr. James Rauschendorfer. And Dr. Rauschendorfer holds an MS and a PhD in forest genetics and biotechnology, where his research focused on plant molecular biology, genomics, and tree development biology. During his graduate work, he helped adapt the fox hunting system, full-length cDNA overexpression, for use in poplar, enabling functional genomics approaches to woody plants. He also conducted extensive research on the role of lonely guy 1, LOG1, in secondary wood formation and the maintenance of the vascular cambium. In addition, his work used RNA-seq approaches to investigate patterns of evolution and speciation in oak species. Following his doctoral work, he completed a postdoctoral appointment at the Donald Danforth Plant Science Center, where he worked on plant transformation in cassava. His work focused on agrobacterium-mediated transformation and plant tissue culture, helping facilitate the development and regeneration of transgenic cassava lines. He currently serves as program coordinator for biotechnology at St. Louis Community College, where he focuses on workforce development, applied biotechnology education, and building partnerships between academic programs and the regional bioscience industry. Welcome, Dr. Rauschendorfer.
All right, thank you. So yes, hello, I'm James Rauschendorfer, and I'm St. Louis Community College's Biotech Program Coordinator. And today, I'm going to talk about a parasitic organism called Agrobacterium. This is an organism that has been adapted in biotechnology to create genetically modified plants. So on this slide, the image that's sort of like shown here is actually one of the plants that I created during my research time using the Agrobacterium transformation system. This is a cassava plant that has been modified to express a green fluorescent protein. So under the right types of conditions, this plant is actually going to glow in the dark. And you can sort of see that glowing kind of going on. If you look, let's just get my laser pointer. If you look right over here, you actually kind of see the plant sort of showing that green fluorescence when you shine the right light on it. Now, today I'm going to walk you through how scientists use Agrobacterium to create genetically modified organisms like the one you're looking at right now. So there's going to be three distinct sections to this talk today. First, I'm going to go over what biotechnology is. And then once we kind of have a good definition of what biotechnology is, I'm going to next explain what Agrobacterium and the crown gall disease are. During this section, I'm going to discuss how Agrobacterium genetically modifies plants and sort of like the use of a small piece of DNA known as the TI plasmid. And then I'm going to end the presentation with my third discussion point, where I will be discussing how Agrobacterium and the TI plasmid are presently being used in biotechnology. I'm going to discuss how this piece of DNA, the TI plasmid, has been altered and has basically been used in what we know as the Agrobacterium binary vector system. Okay, so let's start with just kind of like the first topic. What is biotechnology? So biotechnology is an approach to problem solving. A biotech scientist is one who develops solutions to real-world problems using living systems. And what they rely on to basically make their solutions or make their products are going to be cells, protein, or DNA to help them build a useful product. So on the right is actually an example of biotechnology in action. This is a colony of what is known as Penicillium cryosogenium. And you'll have to forgive me, I'm really terrible at Latin, but the good news is that doesn't really matter because we've all seen this organism before. And you're all gonna commonly know it to be bread mold. Now, what makes this organism kind of important in biotechnology has nothing to do with the way it looks or the fact that it might be eating through some of the food in your refrigerator. It's what this specific type of fungi is actually capable of producing. So what it does is actually really interesting to biotechnology scientists. So let's go over a quick lesson in history. And I need you to go back to like 1928, where we didn't have a good understanding of what different molecules were doing in biology. But there was a scientist named Alexander Fleming, and he was a microbe biologist, and he was sort of like making these plates that he was growing bacteria. And on one of the plates, there's sort of like developed this contamination, and this was specifically a little spot of mold that he was seeing there, specifically the bread mold that was growing. What he noticed when he looked around the site of this contamination is that bacteria would not grow anywhere near where the mold was actually growing. In fact, it looked like the bacteria were like actively avoiding the perimeter around where the mold was forming. And he kind of suggested that maybe this like bread mold was actually causing like sort of like a clear zone or like a kill site where the bacteria couldn't actually enter. So at the time, this was just sort of like a strange observation, and it wasn't actually certain what the bread mold was actually doing that was preventing bacterial growth. Now, let's fast forward a little bit to the present day. Eventually, scientists, they discovered what was something that was actually sort of controlling the bacterial like antimicrobial activity, and they discovered that what the bread mold was doing was it was actually producing the antibiotic penicillin. So the molecular structure of penicillin is actually shown on the right. So penicillin, it interferes with a bacteria's ability to make a cell wall. And without properly building a cell wall, a bacteria cell is just going to keep growing larger and larger and larger until it just sort of like pushes against the cell membrane so much that they just sort of burst open and die. Now, humans, they don't have cell walls. And if a person develops a bacterial infection, they can basically safely consume the penicillin grown from bread mold because it will target the bacterial growth without actually harming the cells of the person with the infection. So everything I just described makes penicillin incredibly valuable. Scientists have, since like the Alexander Fleming's work, have figured out ways to grow penicillin at like a really large scale. And they do this with the help of what is known as a fermentation tank. For reference, the batches of like bread mold that they're growing are going to be between 50,000 and 100,000 liters in volume. So that's a really large amount. Once they actually grow these large cultures of the penicillium microbe, they are going to extract the penicillin from it, purify it, and then package it for commercial distribution as a antibiotic. So at its core, the scientists are essentially growing this like massive vat of bread mold, and they're only taking out the one part of it that's actually valuable. So this is just kind of like the nature of biotechnology. You're going to often be staring at something that like looks simple, something like moldy bread, and then trying to figure out what's actually special about that and how you can use it to produce something incredibly valuable. And in the case of penicillin, it's a medicine that can save millions of lives every year. So now we're going to transition to the next part of the talk, and I'm going to be talking about Agro and the crown gall disease. I'm going to go over three separate things. First, what is a crown gall? And then I'll talk about the organism that's responsible for crown galls, the agrobacterium. And then we're going to spend quite a lot of time discussing how agrobacterium are essentially genetically modifying the plant host to basically sort of develop both a home within the plant that ships food to them. All right. Okay. So just like bread mold we talked about earlier, we are about to follow a similar path in biotechnology. We are going to start with something that looks like a problem in nature and then see how scientists have sort adapted it and turned it into a useful tool. So what you're looking at on this slide is an example of a crown gall. Crown galls are found on woody plants. They can be found on fruiting trees, vines, roses, or shrubs, and they're typically going to form along the trunk or within the root system. These crown galls, when you see them, they're going to have this rough, abnormal texture, and there's a reason for that. A crown gall is actually what we define as like a tumor, and it's a tumor that grows within a plant. The region of a plant where a cell is going to be like dividing is going to be like unregulated, and so that kind of makes the crown gall look rough and swollen and distorted. So these are actually pretty common. So like on this slide right here, we see at least 3 different examples, maybe 4, of crown galls that are on this one tree. We have obviously the really obvious one right here that's sort of like on this branch node. But then we also have at least 1 or maybe 2 seen right here, and then a third that's appearing just over here. So each of these spaces represents a different tumor that is an infection point for the bacteria that we're going to be talking about today. So the bacteria that causes this disease is Agrobacterium tumefaciens. This is commonly just referred to as Agro. Agro is a parasitic organism that genetically modifies plants to make these tumors. And those tumors become both a home that sort of the bacteria live in, but they also have the plants ship food directly to their housing location that they can then consume. On this slide, you'll just see on the right is an example of a few different Agro kind of growing. So let's start by addressing how Agro genetically modifies plants to form crown gall tumor. It all starts with the plant, and here we have a potted plant right now. The stem looks completely happy, but suddenly I would like you to imagine that the plant has been injured. There's a small cut that appears on the stem. Now, normally, when there's a wound like this, it creates an entry point for microbes to infect the tissue. Okay, so it's kind of like getting like a cut. You want to cover it up to make sure that you're not going to be exposing that cut to the like all the germs that are kind of like out there in the real world. So the same kind of situation is also true for plants. But plants aren't defenseless. They have what is known as an innate immune response that sort of helps them fend off microbes. And part of their innate immune response is this chemical called acetylstringone. Acetylstringone is produced when the plants are wounded, and its chemical structure is actually shown on the right. For the most part, acetylstringone is antimicrobial. That means that it's going to help slow down bacterial growth while the plant is attempting to recover from its wound. So shown here, the purple hexagons, they all represent individual molecules of acetylstringone. And what's going to happen is the plant is going to basically release acetylstringone around its wound and basically try to keep the area that's immediately surrounding the wound site sort of free of other microbes that want to maybe infect the branch and get inside the plant. So this is the other character that's going to be important for forming a crown gall. This is Agrobacterium tumefaciens. Agro is an elongated, rod-shaped bacterium. It's small, single-celled, and it's maybe only a few micrometers in length. So that's like maybe almost like 1/500,000th of a meter in size. Its outer surface is smooth, as I've sort of like said earlier, but it's covered in these tiny little hairs that you can probably all see. There's kind of like individual hairs that are kind of coming off. These are just individual flagella that are sort of just allow this microbe to move. It lives in the soil, and so the flagella help to basically propel it through the soil and get it to whatever location it's trying to get to. Under normal conditions, the agro lives in soil where it's going to feed on nutrients that are sort of like deposited by plants or decaying organic matter. But this is an example of an opportunist. It's always kind of waiting for the right moment to become a parasite. And what actually triggers this plant into becoming a parasite, what actually triggers the agro into becoming a parasite is acetylstringone. When the Agro detects acetosyringone, it recognizes that there is a wounded plant nearby, and that acetosyringone molecule tells the Agro there's an opportunity. There's an open point where we can kind of like rush over to and attempt to infect a plant to create a new home and build a new food source that we can consume. So on the last slide, we saw Agro kind of just like acting within soil, not really having like any clear direction, not acting parasitic. But here we see Agro sort of like becoming parasitic in its activity. This is Agro surrounding a damaged piece of plant tissue, and it's aggressively attempting to attach and attempt infection of the plant cell. So they're all sort of shown right here swarming the plant cell, and they're basically trying to find a way to get in and sort of like put their new DNA inside. When the infection is successful, a tumor will start to form. And shown here are like cross sections of plants that are kind of showing how the tumor begins to form. So in A and B, we see like a normal, healthy looking cross section of a plant stem. Go to C and D, and we see where the wound actually sort of like opens up. There's no Agro here just yet. And then in E and F, what you're actually watching is a wound transform into unregulated tumor growth. And that's actually the gall starting to form around where the wound is actually located. So what's happening here is that the Agro is reprogramming the plant cell by introducing new DNA. And this new DNA is going to change the plant cell's behavior and cause them to divide uncontrollably and basically form that gall. So the galls show uncontrolled growth. They continue to expand over time. And in some cases, they eventually will kill the plant. So these can be like pretty severe to damaging plant health. Now, remember, this isn't accidental. Agro is using the tumor as both a new home and sort of like a food delivery system. And the plant is going to be producing nutrients that specifically support the Agro's sort of existence. And it's just basically feeding the infection that's growing inside of it. And all of this is made possible because Agro is genetically modifying the plant. To better understand, we need to talk a little bit about what different types of DNA molecules sort of exist in like the world. And the particular piece of DNA that makes Agro an aggressive parasite is known as a plasmid. Plasmids are small circular molecules of DNA, and they are distinct from the bacterial genomes that we find in the Agro. They're commonly found in bacterial cells. They're rarely found in eukaryotic organisms. In general, plasmids carry what we call accessory genes. These are going to be genes or traits that are not required for basic survival, but it's going to be something that gives an organism some type of evolutionary edge that allows it to survive in an environment better. So plasmids, they're kind of unusual, but they basically just exist in nature as like these completed little DNA molecules. And a bacteria is just able to kind of like swim over them and just sort of grab them and bring them inside. And when they bring them inside, it's going to give them a whole suite of potentially new traits. The plasmid that we're going to be focusing on today is known as the tumor-inducing plasmid, and this is often just called the TI plasmid in abbreviation. This plasmid contains 3 key genetic regions that are important to understand because they are all involved with how the Agro is going to basically act as a parasite. The 3 regions are known as the tDNA regions. This is going to be the portion that is actually transferred into the plant's genome. It's kind of shown here where my laser pointer is kind of pointing to. It spans from one end of this orange region all the way to the other end of this orange region. We then have what is known as the virulence genes, which are shown as kind of like a gradient of pink to blue. All of these are basically little toolkits that help us ship the DNA into the plant. And finally, we have over here in yellow our opine catabolism genes. These are all going to be responsible for like basically allowing the Agro to eat the food that the plant is making. So let's focus on each of these regions just sort of one by one and sort of talk about what's going on. So first, we're going to look at the tDNA. This is what contains the genetic information that the agro is actively trying to put into the plant. This is the part of the that the agro is going to be genetically modifying the plant with. And there are three key genes that are all found within the transfer DNA region. The first is an auxin-producing gene. So for those of us that aren't really familiar with like botany, auxin is a plant hormone that regulates cell elongation and division, and it's especially important in growing tissues of roots. And in the cartoon that you see on the lower left, what we're showing here is auxin, the molecular structure, and it's being added to a plant cell. And when it's added to the plant cell, it causes that plant cell to basically grow, divide, and make two daughter cells. So each of those cells that come from the cell that was given the auxin, they are all going to contain genetically identical genomes to the cell that was just divided. So this is the first part of how Agro introduces this is the first gene that Agro is going to introduce in the plant's genome. And when the gene is expressed at high levels, it's going to drive excessive cell growth that is going to contribute to tumor formations by basically causing that unregulated division. The next part that we're going to look at is the cytokine producing gene. Cytokines are just another type of plant hormone. They specifically stimulate cell division, and they also help maintain the dividing state. So the cartoon at the lower left, again, kind of looks very similar to what we're looking at with auxin, but instead of auxin being added this time, we have the molecular structure of one type of cytokinin that's being added to the cell, causing the cell to grow and divide. Again, when the cell grows and divides, it's going to have genetically identical genomes that are in both of the plant cells. So both auxin and cytokinins are present at high levels because these are going to be actively expressed at high levels when we introduce this into the genetically modified cell. And they're going to unregulate cell division and really start the process of growing that plant tumor. The final part that we need to talk about is known as a gene called the opine synthesis genes. Opines are just specialized molecules that basically serve as a nutrient source for agro. They provide a rich source of nitrogen, which is the limiting resource that is often not found within the environment. The opine synthesis genes are essentially like a recipe that the agro forces the plant to follow, and it's directing the plant to produce a specific food source right at the tumor's location. Okay, next on the list of the gene regions we need to talk about are the virulence genes. So virulence genes are responsible for making the tDNA and moving it into the plant genome. So they are literally going to take this region up here and completely shuttle it into the plant's genome. So that's this region here. Virulence genes shuttle the tDNA inside. This is a very complex process that's going to involve a set of genes named Vir-A through Vir-H. They're all shown kind of right here in the order that they appear on the virulence gene region. And each is going to produce a specific protein, and all of these proteins are going to work together to help transport the tDNA from the agro into the plant cell. And we're going to spend quite a bit of time talking about how these genes operate with agro to get this done. Now we're going to shift a little bit, and we're going to talk about how all the virulence genes are working together to move the tDNA in the plant. And this is going to be kind of like a long series of cartoons that shows how the step works, step by step. We're going to start in agro, which is shown here in kind of the peachy color, and eventually we're going to watch the tDNA make its way inside of the plant. Okay. So the first two virulence genes we need to discuss are Vir-A and Vir-G. Vir-A is what is known as a membrane-bound receptor protein, and it's embedded into the agrobacterium cell membrane. That means that part of Vir-A is sticking out into the environment, and it's acting as sort of a detector to find a very specific molecule. Vir A is basically searching through like soil to find acetostringone. Because when Vir A finds acetostringone, that's it detecting a signal from a plant. And specifically, it's sensing the presence of acetostringone, and it tells the agro there's a wounded plant nearby, and we can start to like act aggressively as parasites. So Vir A binds to acetostringone. This tells agro that the wounded plant is nearby, and it's going to signal the cell that it's time to initiate the switch to the parasitic state. Essentially, it's the message saying it's time to start actively trying to infect. Now, the signal needs to be moved from the outside of the cell to the inside of the cell. And this is done by relaying the message of Vir A onto Vir G. So Vir G moves through the cytoplasm towards Vir A. And when it binds to Vir A that has sort of detected acetostringone, it is activated. So Vir A in this state is going to be transferring a signal to Vir G through what is known as cellular phosphorylation. This phosphorylation is going to activate Vir G and turn it into its active form. And in its active form, Vir G is then going to swim all the way back to the TI plasmid, and it's going to carry that simple message. It's telling the TI plasmid, hey, we found a wounded plant. It's time to infect, and I need you to start producing all the virulence genes to make this happen. So Vir G binds to the virulence gene region, and it switches on the virulence genes of the TI plasmid. Once bound, Vir G is going to turn on expression for all the genes here. So the first two genes that are going to get expressed are Vir C and Vir D, and these two are going to be expressed together. Vir C, it's going to bind to the borders of the tDNA region of the TI plasmid, and it's going to basically establish the region that's going to be moved into the plant. So it's defining where the start and stop of this DNA exists. And it's going to be positioning it here so that way we can correctly transfer this region. It's marking the exact segment of DNA that's going to get moved over from agro to plant. And this sets the stage for the next step, where the tDNA is going to be cut out for delivery. So now the tDNA has been sort of marked, the Vir D protein moves in, and it's going to swim through the cytoplasm to where Vir C is bound, and it's going to snip the tDNA region out of the TI plasmid, and it's going to create a single-stranded piece of DNA that contains all of the tDNA. So it makes precise cuts at the border, and this is going to release the tDNA from the TI plasmid, so now it's going to be transferred over. Vir D is going to remain attached to the sites that it cuts, and the reason that it remains attached is because Vir D, in addition to cutting out the DNA, it also kind of acts as like a pilot that guides the tDNA into like the host plant cell. So it's going to be basically driving the tDNA over from agro to plant. Now, the next virulence gene we need to discuss is Vir E. Vir E acts as a chaperone protein. It's essentially kind of like a bodyguard that's going to protect the tDNA. And the way it protects it is by basically coating the tDNA, and it's going to make sure that when the tDNA enters the plant, it's not going to get like broken by the plant's defenses. This protection helps keep the tDNA stable and prevents it from being degraded while we're transferring it over. So we literally have Vir E coating the tDNA, and Vir E is kind of like the bodyguard, and it's going to be protecting the tDNA from the plant. So now the tDNA has been prepared, and it's time for the agro to deliver the tDNA into the plant cell, which is shown here in green. And ultimately, it's going to want to end up in that blue region, which is just kind of representing the plant's genome. To facilitate the movement of the TI plasmid from the agro into the plant cell, we're going to be relying on Vir B. Vir B forms this channel that basically allows agro to ship things from the agro cell into the plant cell. It's more or less, Vir B, it's like a tunnel that allows tDNA to basically move directly through and get to the other cell side. So shown here, Vir B is forming a connection between the agrobacterium and the plant cell, and the tDNA is going to use this like tunnel to move across the boundary, and it's just sort of bridging that gap from bacteria to plant. But before the transfer can happen, the plant has more defenses. It's not going to just accept foreign DNA without fighting. It's actually going to be actively producing things that want to kill off the agrobacterium and destroy foreign DNA. So the plant is actively hostile to agro. It's basically producing all these antimicrobial compounds that mount this very large defense response and targets both bacteria and any incoming DNA. So under the normal conditions, this environment would prevent agro from surviving and would block the tDNA from being transferred. However, agro kind of has like an Uno reverse card called VIR-H, and it's just sort of shown here with a smiley face. VIR-H is going to be delivered into the plant cell through VIR-B, and what it's going to do is it's going to neutralize the plant's defense, and it's going to basically dampen the signals where the plant's screaming that something's wrong, and it's going to make conditions favorable so that way infection can actually happen. So VIR-H is basically going to move into the plant cell, and it's going to tell the plant, hey, everything's fine, just ignore everything that's going on around you. So VIR-H moves across VIR-B, and it turns the plant into kind of like an agreeable host to just sort of take on this new parasite. Now begins the production of VIR-F. VIR-F is going to travel with the tDNA through the DNA cell all the way into the plant's genome. And they're going to move together and kind of like pass through the VIR-B channel into the plant. Inside the plant, the tDNA complex is going to move along with VIR-F, and they're going to move into the nucleus shown here as blue. Once inside, the tDNA is going to integrate in the plant genome, and this is where genetic modification occurs. This is like where we're actually changing the plant's genome to behave the way that the agro wants it to behave. So inside the nucleus, VIR-F first removes those chaperone VIR-E's, and this exposes the tDNA. Next, VIR-D sort of just disassociates from the tDNA, and now we have this sort of like free single fragment of DNA. And the plant actually can right now not tell the difference between the tDNA and its own DNA. And it's just sort of assuming that like this is a fragment of DNA that needs to get integrated into itself. So using the plant's like own DNA machinery, the plant is essentially going to stitch the tDNA directly into its genome. And here we have it. The tDNA is now directly woven into the plant's genome, and now it's possessing genes for inducing tumor growth and producing the agro's food. So everything that we just discussed is a single cell in the plant that contains the tDNA that is now primed for tumor formation. And so just as a reminder, we have what the transferred DNA actually looks like up the top, shown here, that we'll be referring to for a little bit. Now, the infected plant begins uncontrolled cell division. This is driven by the auxin and cytokine producing genes that are carried in on the tDNA, which are going to push the cell into continuous growth and cell division. So when you're handing over these two hormones to the plant cell, what will happen is the plant cell is going to divide and replicate their entire genome. That means that the cell containing the tDNA will make copies of itself, and every new cell is going to carry the exact same genetic information from the original copied cell. So every cell made after this cell division is going to be a replica of the genetically modified cell, and they'll all carry that tDNA. So the cell divides first. One cell becomes two. 2 cells becomes 4, 4 cells becomes 8, 16, 32. It grows at an exponential pace, and this leads to an unregulated tumor growth in the plant. So on this slide is a sketch of what a gall tumor looks like. The green mass represents the tumor itself. All of the cells on the gall tumor contain the tDNA, and now there's still one of the tDNAs that we haven't genes on the tDNA we haven't discussed yet, and this is the opine synthesis gene. So this is the gene that causes tumor cells to produce opines. And here we have it. The tumor cell has now produced a bunch of the opines. Okay, so now here we are at the tumor-inducing plasmid, and we're ready to talk about that final piece. We haven't talked about that, and that's going to be the opine catabolism genes, shown right here in yellow. Agro has specifically involved the ability to basically use opines because it contains the TI plasmid. So this is kind of like a really kind of like twisted and sick part of it all. Like opines are not normally like a common food source for microbes. Like microbes don't eat these at all. The only reason Agro wants to eat these is because it carries the tumor-inducing plasmid, which has both the gene for making opines and then the gene for breaking them down as a food source to feed itself. So essentially, this is just kind of like one of those strange things that's really kind of indicative of this plasmid and its parasitic function. So now we have a gall tumor. This is going to become the Agro's new home. So that's the part shown here in green. And the tumor is producing opines, and they're all going to become the Agro's food. At this point, the Agro just simply needs to move in and colonize the tumor. So shown kind of like those peach kind of like ovals, those are our Agro. And all that happens next is they basically just move inside, and this is where they live and reproduce and just sort of grow with just a food source provided to it continuously. Okay, so we spent a lot of time discussing how this works. Now we're going to talk about how Agro is actually used in biotechnology. We're going to talk about why do scientists genetically modify plants. Then we're going to discuss how the TI plasmid has been altered to make it no longer responsible for parasitism. And then we're also going to discuss sort of like how the binary vector system is used for making a, I'll think of the word, genetically modified plant. Okay, so scientists are going to perform genetic modification as a way to introduce a specific trait into a plant in a controlled and targeted way. This allows a scientist to improve characteristics like yield, disease resistance, stress tolerance, or nutritional value without relying on sort of like random variation or kind of like the difficulties of like plant breeding. Compared to like traditional plant breeding, genetic modification is way faster and it's much more precise. So instead of crossing plants over multiple generations, hoping that you're going to disappear the traits that you don't like and then sort of like prop up the ones that you want, genetic modification allows you to directly introduce exactly what you want into the plant. One of the key advantages of using Agro is that Agro is already kind of like primed to infect many different plant species. It likes to infect trees. It likes to infect like fruits. It likes to infect vines, and roses, and shrubs, many sorts of things. So this is kind of like becoming like this universal tool, or maybe even like a workhorse that scientists can basically use to put a fragment of DNA that they choose into a plant. So let's go back to the TI plasmid. This is an incredibly powerful piece of DNA, as I've kind of like already shown you. And it's capable of changing a plant's entire genome and modifying the way that the plant behaves. It's what allows Agro to genetically modify plants. We are now going to shift gears and look at how scientists have taken advantage of the system to create genetically modified plants. And this is where biotechnology sort of like steps in and sort of redesigns the TI plasmid for more controlled use. So the first step in redesigning the plasmid is removing all the parts that we don't want. Specifically, we are going to, as scientists, remove all the parts from the plasmid that make agroparasitic. All we want is to keep the components that allow us to genetically modify plants. So first, let's just start with the tDNA region. This is like a portion that's pretty integral because it actually takes a fragment of DNA and integrates it into the plant's genome. Biotechnologists, they want to keep the tDNA's ability to transfer over, but they don't want to carry all those genes that are going to cause tumors or are going to like be related to like make strange opine food source. So This region is edited by removing all the auxin, cytokinin, and opine synthesis genes. And what's left is this clean genetic shuttle that can be transferred to the plant, but it no longer carries the tumor-inducing genes. So all of those genes that I just discussed, they're removed. Now, this gets us on the right track, but it doesn't give biotechnologists the ability to basically transfer the DNA that they want to see inside there. This is just sort of like an empty space. So we want to sort of like improve this region so that way it's going to carry useful genes over. So the tDNA region is then further redesigned to introduce what's known as a molecular cloning site. Okay, so a molecular cloning site, this just means that it's really easy to basically take any piece of DNA from any organism that we want related to any trait, and we can basically put it into this region. So right here, this part that's like sort of marked in red, that's going to be our multiple cloning site. And scientists can basically take whatever they want and put it directly into this location. Okay, the next set of genes that got to go are the opine catabolism genes. This is the part that's marked in yellow. These genes serve no purpose for genetic engineering. They only support the parasitic lifestyle of Agro. So in this system, what happens is they were just simply removed. And now they're gone. Now, the final part left to discuss is the virulence genes. Virulence genes are essential for transferring tDNA into plants. They provide the machinery that makes the whole system work. They basically are going to take that tDNA and get it over. So this is a portion of the TI plasmid that we want to keep fully intact, and we don't want to modify this at all. We are going to keep the virulence genes as they are. Okay, so now we're going to do something that might seem a little bit odd, but I promise will make sense once we We're going to take these two different gene regions, the virulence gene and the tDNA, and we're going to basically move them onto two different plasmids. This is called the binary plasmid or binary vector system. So we're going to have two different plasmids. The virulence genes are kept on the plasmid known as the helper plasmid. This is the plasmid that helps the tDNA get into the plant. And then the tDNA is going to be kept on a different plasmid that's just known as the binary vector. So why are we splitting the system up? That would be a good question to ask. We separate these components, We separate these components because only the tDNA is the part of this plasmid that needs to be genetically modified. By keeping virulence genes on a separate plasmid, we're basically making sure that we're not going to disturb the virulence genes at all, and we're only going to be editing things on the binary vector. So we're going to just kind of make it only manipulating the binary vector. So let's talk about how these plasmids are sort of like used individually. Oops. That laser pointer is annoying, sorry. So first, let's just start with the helper plasmid. This plasmid contains all the virulence genes, and it's going to provide the machinery for getting tDNAs across. There's no need to modify this plasmid at all, so we're just going to keep it and store it in the Agro. So this plasmid is just going to live inside of this like new Agro strain. It's going to have none of the genes that are going to make it parasitic, but it just contains the virulence genes. So it's maintained inside of our agrobacterium. So now this redesigned Agro, it's going to have all the machinery parts required to basically allow for genetic engineering to happen. The only thing it doesn't have yet is the DNA that we want to deliver into the plant. So now let's jump back to the binary vector. The binary vector contains the modified tDNA region with a multiple cloning site, and this is where a gene of interest can sort of like be inserted into. We're going to use this technique called molecular cloning to insert a gene of interest into the multiple cloning site. And we actually don't have enough time to cover how molecular cloning works today. The process is a bit complex, and it's something that can be performed really easily and routinely in the lab, but it just takes quite a good amount of training. And you'll just have to trust me that this method allows us to insert a gene from any organism with any trait we want to represent and just introduce it into a vector system. Now, if you join the biotech program, You'll actually learn how to do this in Biotech 1, where we actually spend quite a lot of time with molecular cloning. So that's just a pitch for my program. But anyways, so now the gene that we want, the gene of interest, it's been put into the tDNA and it's ready to be delivered into the plant. And this is done by mixing the agro and the binary vector in sort of like a solution together. And right now the binary vector is outside, but they're both kind of swimming around together. And then we're going to just sort of like pulse this briefly with a little bit of electricity. That brief electrical pulse is going to temporarily open up the cell membrane of the agrobacterium, and it's going to allow the binary vector to basically get inside. So here's the electricity striking the cell, and the binary vector just jumps right inside. Now, the last thing we have to do is mix this transformed agro that we sort of like have added both the binary vector and helper plasmid to. We need to mix it with both plant cells and a little bit of acetosyringone. And the acetosyringone is going to bind to VRA, activating VRG, which in turn is going to allow for the shipment of a tDNA into the plant. So we're going to cut the tDNA out of the binary vector. We're going to attach our two VRD genes to either end to be the pilot and sort of escort it over with the VRE chaperone, create the VRB, VRH, and VRF, shuttle everything across into the plant cell, and ultimately, we're going to integrate the tDNA into the plant cell, and we've now created a genetically modified plant cell. And you can take the cell and grow it into a full-size plant through techniques of tissue culturing and transformation, which we don't really have enough time to talk about just today. Okay, so now if any of these topics interest you, I want to point you towards the Biotech program. If you want to learn how to generate a fully genetically modified plant using the method that we just talked about today, I'm going to suggest that you enroll in our Summer Advanced Topics course, BTX 226. This will actually be covered in this iteration of the course. I'm going to be sort of onboarding a professor to sort of like teach about this today. And he's going to be basically helping me build a class. I believe he's going to have you all make a genetically modified potato that has the green fluorescence protein. So you'll get to make a potato that glows in the dark. We'll see. I think that's the direction he's going. And if you're interested in how biofermenters look, like the example that we talked about with penicillin, then I'm going to suggest you take the advanced topics in the fall, where we'll be developing a whole course around biofermenters and carbon sequestration. So basically stealing carbon out of the atmosphere and locking it away so it doesn't contribute to global warming crisis. If you'd like to learn more about molecular cloning, so basically making your own plasmids and re-engineered DNA, then I want to suggest that you register for Biotech 219, Biotech 1. We spend about half the semester learning techniques to create genetically modified bacteria. And we've even had a few successes sort of like in this semester. I want to call out, shout out to Ricky Lane, who's on this call. He successfully made a transgenic bacteria with a correctly redesigned plasmid. So great job over there. And all these courses are offered through the Biotech Associate's degree or certificate program, which are designed to prepare students for jobs in the biotech industry here in St. Louis. So if you're interested, feel free to reach out to me. Here is my contact information. Thank you for attending my talk. Please email me if you have any questions, and I can take any questions that you have on agrobacterium right now. We'll see. I'll do my best to answer them.
Thank you so much, Dr. Rauschendorfer. If anyone has a question, or feel free to put them in the chat. I have a question for the galls that we see on plants, whether they're on the trunk or in the roots. How long does it take to kill the plant or the tree? Or is it always fatal?
It is not always fatal. It's kind of like the location of the gall determines if the gall is going to be fatal. So maybe I should kind of like go back through the PowerPoint. Sorry. I got a million slides. Sorry. Let's just go to the top. Okay, so yeah, it really depends on the gall's location. So like right here at like the root, that could be like, if it's the junction point between root and the stem, that could be a pretty critical point because it's basically going to choke out like where the flow of nutrients goes. If it was more kind of like farther away on the root, that's not a big deal. Eventually, that's just going to kind of like that tissue will die off and they'll leave it alone. If it's on a branch, not a big deal. But if it's on the main trunk, then you truly have a problem. There's actually quite a few examples of this. If you walk around Tower Grove Park, there's some really interesting bald cypresses that you can see. I really wish I got a picture of them, but there's some really bad crown galls on the base of a trunk of a bald cypress in Tower Grove Park, and that looks like something. I mean, it's been growing for a while, but it looks like it might be something that could kill it in like 10 years. Yeah.
I do see as I walk around my local parks, usually
Mhm.
smaller galls on, and they seem to fall off attached to the branch. Is that the same thing as these larger galls?
Ooh, so that's a different genetic modifier, most likely. I think it might be a different one. So wasps also like to be genetic modifiers, and they can use like different mechanisms with like viruses to like basically make like gulls that they can sort of like let their sort of like offspring grow inside of. So sometimes it really depends. Like I feel like the wasp galls are much smaller. There's some really good examples of these on some of the oak trees near the they're on the Merrimack campus of St. Louis Community College. And you can kind of just see them kind of like growing on like the really like tiny branches. That would be I think those are wasp galls. But yeah, wasps can also genetically modify things.
Okay, here's another question. Have we engineered a version that is compatible with animal cell delivery and modification, or is that dedicated to other technology?
So agro only works with plants, but there are other like systems for genetically modifying plants, and I really don't know them all that well. They're not really like my forte. I spent my whole like grad school like basically studying how to make genetically modified organisms, but I only know how to work with plants. So like learning how to like sort of make genetically modified animals just not something that ever took up my interest. I'm certain it's pretty similar. I believe you kind of have to use things like horizontal gene transfer, and I don't know, CRISPR technology is really kind of just like revolutionized like really how we do everything in biotech. So it's really easy to actually make genetic modifications in pretty much most systems. If there's a way to do it, then like CRISPR is going to make it kind of like super easy to like make it do what you want.
Any other questions for the chat? Okay, well, Dr. Rauschendorfer, thank you so much for your presentation today. We learned a lot about biotechnology and agrobacterium, and thank you once again.
All right. Thank you. Have a good day. Hmm.